I'm Autumn Malloy, and I'd like to welcome you to the Westmoreland County Community College Microbiology Lab. I thought I'd give you a quick tour so that even though you're online, you can still see some of the features we have. And we're going to be focusing on the storage and biohazard disposal sort of things today. So one of the first things as you come in the door, you're going to see over here, we have our eye wash area. We can see we've got an eye wash here. Now, if you've never used an eye wash fountain, the way this works is you pull this down and you see the water rushes up. And if you had chemicals or something in your eyes, you would put your eyes in there for 15 minutes and let that water flush them out. When you do that, you want to be careful to not have any contacts or any glasses on, because if you have contacts in, that water will force the contacts up into your eyes. We also have our first aid kit up here with the standard bandages, burn cream, smelling salts, things of that nature. We don't usually have to use that. Occasionally, somebody will get a cut or a burn during microbiology lab. But usually, if we use it, it's because somebody has a blister on their foot and they want to cover it up from their shoe rubbing on them, or if they've got a cut and they want to cover it before they start working with bacteria. In addition, in this same area, up on the ceiling, we have a fire shower. Now, I'm not going to demonstrate that for you, but if I were to pull this handle here, what would happen is water would come flooding out of the ceiling, and if you had a big spill or a fire, that water would come down and flush you out. Now, I've never had to use the eye wash in microbiology, and I've never had to use the fire shower in microbiology. I have had to use the fire shower when I was teaching organic chemistry, but never in microbiology. When we look at the area, one of the things that's important is to keep the floors free of clutter. And so we always have cubbies for students to put their books and book bags and coats and everything in. Here we have the microscope location. And we'll be looking at the microscopes later on here. If we look at the desks, one of the things that we do is when a student first comes in, they're going to use back down on their bench. And that back down is a solution that's antibacterial, and they will saturate the bench and wipe it off, and that will kill any bacteria or viruses that are sitting on the bench to prevent your work from getting contaminated. Then, at the end of lab, you will also use back down so that you're killing off anything that's left behind. One of the reasons that I've never had to use the fire shower in microbiology is because we don't use flames. Instead, on the benches, we have these devices, which are called bacti-incinerators. If you turn them on, what happens is there's a ceramic core inside of there that gets red hot. And as the core gets red hot, if you have a tool that you're using to sample microbes, you can put it in there and the tool will get red hot. And once it gets red hot, it is hot enough that it will kill all the bacteria and viruses and fungi and everything, and so it is sterile at that point. As we slide around, also on the bench tops, we see these small orange disposal bins. And these are at every bench station, and we put used slides or used toothpicks or used cotton swabs or anything glass that's being disposed of goes in there. One of the things that we always use is gloves. And so we have a whole array of gloves to be used. As we slide down and we walk further around the lab, we see one of the features that we'll be doing experiments with later on. This is one of our chemical hoods. Now, with the hood, we have this exhaust fan, so you can hear it when I turn it on. Air comes through, and if any fumes are generated, they'll be kept in this hood and taken out the vent. But one of the main things we use it for is this feature, the UV light. If I go ahead and turn it on for you, you can see that it generates that characteristic bluish purple glow that we associate with UV light. And we'll be using that in different experiments to test the effectiveness of UV light against different bacteria. So we'll go ahead and turn that off for now. Further on in the lab, we have our incubator. And you can see that it is set for 37 degrees Celsius. Those of you in the allied health field might know that 37 degrees Celsius is body temperature. The reason we have it set to that is the temperature at which most organisms that we use grow. And so the incubator is used for growing bacteria. And if we open it up, we can see that some of our classes have bacteria in it growing. Now, if we have bacteria that have already grown and we put them in the incubator, one of the things that can happen is something called overgrowth, where the bacteria will continue to grow and continue to grow, and they run out of nutrients, they produce wastes, and they will actually die. So, if we have bacteria that have already grown and we want to keep them, one of the things we'll do is we'll keep them in this room temperature cabinet here. And so we have the incubator for growing bacteria, and we have a room temperature cabinet for storing bacteria. As we progress through the microbiology lab, another feature we have is the bulk disposal. And so this is a bin that we put in large things, petri dishes, paper towels that have been contaminated with bacteria, any gloves that might be contaminated with bacteria, things that won't fit in the small little disposals on the bench tops. As we come around, here we have our sterile area. Everything here is sterile, which means it doesn't have any bacteria on it. So today, we can see we have some petri dishes, we have some test tubes, we have some sterile swabs, and that's what they were using in their experiment today. So, as we progress around the lab, you can see from the back of the room, this is what it looks like. So if you were a student, you'd see me up front with all the boards, all our plants. We have plants everywhere in the lab here. We have refrigerators that are used to store bacteria and media so they don't go bad. We have another hood, and then we wrap up with more microscopes, more cubbies, and a fire extinguisher. And again, I've never had to use the fire extinguisher because that's not one of the big concerns we have in microbiology lab. In microbiology lab, the big concern we have is contamination. which is why we use back down and back the incinerators and gloves and try our best to keep our area as free of contamination as possible. So I hope you enjoyed your quick tour of the microbiology lab and some of our safety features. I'll be talking with you again and throughout this semester.